I hope your parents signed your permission slip because today Plants in Jars is taking you on a field trip to the University of Florida's Ornamental Tissue Culture Laboratory. If you've never seen me before, hello, my name is Lore. I make videos about tissue culture, which is a scientific process of mass-producing plants that are genetically identical. It's very cool. You can do it on any plant. This video is going to cover 5 research topics: Monsteras, ghost orchids, wasabi, cryopreservation, and hemp, which is the most requested plant on this channel. Before we embark on our field trip, I have one final announcement, and that is that I have merch. I worked really closely with the artist on this design. The front has a jar that says Plants in Jars, and then the back has a really awesome skeleton doing tissue culture. I'm so proud of how it came out. Buying a shirt is a great way to support the channel and the educational content that I make. Okay, let's get started.
Hi everyone, my name is Vendrame, originally from Brazil. Vendrame is an Italian last name, so you know a little bit of my background. I am a professor here, but also act as interim department chair for the environmental horticulture department. I've been with the university for 23 years now, and most of my work has been on plant tissue culture and micropropagation. We work with ornamental plants, food crops, and also some medicinal crops.
Wow. This is awesome. So up top you guys mostly have the Monstera type constellations, it looks like. I think I read in your research paper you guys were using 8 milligrams per liter of PAP.
Yes.
To me that seems really high. How did you guys end up figuring out that hormone combination?
We started with low concentrations, but we were not getting any results. So what happened is that we had to adjust. So we started going higher. So we look at 7.5, 10 milligrams per liter. And then we realized that the 10 was too high, but 7.5 was the level that we were looking at. And then we end up fine-tuning and settling on the 8. With that, we were able to get good multiplication. With the lower levels, we will get one shoot to grow, but we wouldn't get multiplication, multiple shoots. For every species, each species is different, and even within the same species, if you have different cultivars, you have a genotype effect. Yeah, so each plant has its own genetics. So basically, you need to make adjustments in your culture medium. You can start with a basic medium like MS medium, and then with the plant growth regulators, we usually start with the literature search. So we see what's in the literature. Has anybody done any work with this species? If so, that's a good starting point. We already had some reference there. But if there's nothing, then we go to the genus. So let's say we couldn't find anything on a specific, some Dendrobium species. So we go to the genus Dendrobium and see if there are works published with other Dendrobium species. Again, it gives us a good start because it's the same genus. And from there, we develop our own protocols, or we do research. So part of the research is that, okay, we determine a culture medium. Now let's see what are the parameters that we can evaluate to improve multiplication. So we look at, let's say you have an auxin, which is a plant growth regulator, and you have a cytokinin. So we start playing with the different levels, and you can make a matrix. For example, 3 levels of auxin, 3 levels of cytokinin. So it's 3 by 3, you're gonna have 9 treatments that you're gonna look at. And among those 9 treatments, you do repetitions. So you have maybe 10 units per treatment. So after you evaluate those, we do some statistics to see which ones are really working best and show the best growth, best multiplication, best quality of plants, best rooting, anything. And that's when we determine, oh, this is the best level. And that's how we got to the 8 milligrams per liter for Monstera. And then we move it into the bioreactors because we thought, well, if you want to improve multiplication, here in the jar you may have maybe 7, 8 shoots, 10 pushing it, but the space here is limited. So why not to use a larger container? So there are different containers you can use, like you can use a magenta box that's slightly bigger, but not much bigger than a baby food jar. And these are clerodendrons, is another ornamental. Or you can use the bioreactors. Right now we don't have anything growing on them. When you look at this jar, what do you have? A culture medium and a plant, your ex-plant. The culture medium has the nutrients, the plant growth regulators, but it has agar. Agar is the gelling agent, like think jello, right? So you have plants in a jello full of nutrients. The bioreactor concept is basically the same thing, except that you don't have agar, the gelling agent. You have liquid medium that sits here on the bottom, and on the top you put your ex-plants, your little plant tissues. And then you have a compressor that pushes compressed air into the system. It pushes the medium from the bottom container to the top container. This is called immersion. So because the tissue get immersed into the liquid medium, so we can control these two parameters of immersion, frequency and duration. And we can also inject air through this line to the top to refresh the air in where the explants are, so you get like a renewal of air for the plants to grow better. So this system is very efficient. As you can see here, you can produce like hundreds of plants in a little box like this, sometimes thousands. I give an example with sugarcane. It was the first thing we tried. We started with 80 little shoots, this size, 80. By the end of 9 months, we have about 1,000 plants.
Wow.
So the rate of multiplication is over 10 to 1. And that's what you want. And that's what makes the system efficient and cost efficient. Because think about it, now we're not using agar. Agar is expensive.
Yeah.
It's probably 80% of the cost of tissue culture is by the agar. By eliminating the agar now, we're making it a little cheaper. And because we produce a lot of plants, the cost per plant is reduced. You could get maybe a plant here, each plant will cost you maybe 50 cents each. When you sell a plant for $1, $1.50, that's a good margin, right?
Yeah.
Ornamentals are always in demand, especially when the Thai constellation, when I learn about the Thai constellation, you will go to Etsy and you will find a plant that will cost $2,000.
Yeah.
There are people selling a Monstera in a pot like for $10,000, and like that's insane.
There's this one really cool one called the Kunzo White Monster, which is a white variegated Monstera from Japan, which is like it's super hard to get. I've been trying to buy one, but the cheapest one I can find is $700.
Wow. If you get a plant like that costs you $700 and you're able to multiply, just think you produce 100 plants. Look at how much money you can make. Even if you sell them at $300 instead of $700, do the math. $30,000.
Well, now I know 8 milligrams per liter of BAP. And you guys have some of the ghost orchid seeds growing, right?
Yes. You know, most orchids are considered endangered or threatened. So there is a lot of effort in preserving them. The ghost orchid was like controversial maybe in the 90s when a guy came down to the Fakahatchee strand and he tried to poach these orchids thinking that he could clone them and make tons of money. But that's considered a federal crime, so he was arrested. And Susan Orlean, a reporter from the New Yorker, came down to South Florida, and she was puzzled that how come somebody will go to that extent to steal an orchid and get arrested for it. She was curious about it, and she wrote the book, The Orchid Thief, that goes, all about the ghost orchid. So the ghost orchid became an icon in Florida. It's called the ghost orchid because it's a very tiny orchid. The flower is about this big, but it doesn't have leaves. It's a leafless orchid. So the roots, they spread and they attach to the trunk like this. So it looks like an alien creature attached to the tree, but it's a bunch of roots. And it has a little growth point, a very reduced stem that you can barely see. From that stem, it comes the flower stalk. And usually when they are in the trees, the flower stalk comes this way, and the flower is white with some appendage, and it swings when the wind blows. So it looks like a little ghost.
These are the seeds of the ghost orchid.
Yeah.
So when you see these little green structures growing, those are called protocorms. They look like little round green structures, just like peas. And those are the initial germination stage of an orchid seed. Then from that green structure, the protocorm, you start getting little leaflets forming and root primordia. And then eventually they start elongating and forming a little plantlet. Now orchids some can be difficult to propagate because basically they're in the grass family. So they have one meristem, and if you kill it, basically you kill the plant. So you need to work with a variety of explants. For example, when we started working with orchids, we started with Phalaenopsis because they're readily available. You find them everywhere. We knew that if we use the meristem, we will kill the plant. Or if we use leaves, it will be a little more difficult. So we started working with flower buds. When the flower spike comes up and has the little flower buds that haven't opened yet, that tissue is highly meristematic, which means the cells are actively dividing, and that's a perfect tissue to put in tissue culture. Another way to start is to get these little green structures, the protocorms. That tissue, that seed that is growing, the protocorm, is like a meristematic tissue also, is actively dividing. So you can kind of cut in little pieces and you can induce the formation of those embryos, and they will form little round structures called PLBs, or protocorm-like bodies.
Okay.
And those will turn into plants. So from one little piece you can have maybe 20 PLBs, which means you could have potentially 20 plants from one little tissue.
Wow, okay.
So that's one way of multiplying orchids too.
We have similar material to that in here. It's not from orchids, but it's from the saw palmettos. And these are grown from callus, which we started from flowers. It was the same basic process, but it was with saw palmettos instead of orchids. And you can see in here, it's just sort of random cells which are starting to form into new plants. There's really not much to them, but if you treat them in the proper conditions and with the proper growth regulators, they will turn into whole new plants, which I don't see many here, but there are a few beginning to form embryos. This is required to be locked. This is hemp.
Oh.
And you can see they're growing fairly well. No one really has a published, well-working method for the tissue culture of cannabis yet. Many people are trying it, but they are not getting very good results. We seem to have a fairly good method, as you can see, and they grow very vigorously, but the problem we've been having is they don't last very long in culture. The media runs out of nutrients extremely rapidly, and it causes extreme chlorosis in the plants, and they have to be transferred at probably every 2 weeks would be best.
Wow, that's a lot of work.
That's why we don't work with a lot of them.
It's a very tricky culture. Even within the same genotype, the same variety, if we take different parts and we put them, they start behaving differently. So it's a plant that seems like it varies very quickly when you put in vitro, and it consumes a lot of nutrients. So it's like they're very vigorous plants, but they're actively like changing.
Yeah.
So that's why I think it's difficult for people to develop a successful protocol.
Yeah.
But we've been working already a few years with it, and we're getting good results. But again, we have those issues. At some point, we had some hyperhydricity too, right?
Our last experiment, which we just took out of the bioreactors, one of the issues we were dealing with was trying to remedy the hyperhydricity. We were able to get a fairly good reduction in the hyperhydricity, which is just done through altering the ventilation cycle. In the bioreactors, hyperhydricity can sometimes be hard to control because you're growing it in pure liquid. You can see sort of the result of lacking ventilation here. These you would not consider to be very good looking plants. These are potatoes, regular solanum, and you can see they're sort of all malformed. This one is more of what it should look like. The reason they look this way is because the caps are sealed and it prevents enough fresh air exchange. When solanums don't get enough fresh air exchange, they seem to suffer from ethylene buildup, and ethylene is a plant growth regulator, but all plants give it off. It's a gas. And so if the container is sealed too tightly, the plants can't really grow properly because the ethylene is altering their growth, which is why they're not really producing many leaves and they're just all callous.
I'm working on Wasabi. It's hasn't been studied very much in different areas of tissue culture. It's having some problems because of climate change in some areas. So it's really important that we keep the species, but right now I'm just trying to get the plants, start the process of getting them in vitro and then hardening them off and putting them off into a different area.
Sweet, that's so cool. Oh yeah, they are like completely white. Wait, why did that happen?
So there's a big problem in Canada. It's called blackleg. It's a fungus in the soil. So when I got these plants, because it's only grown in like 2 areas of the United States, Washington, Oregon, and then in Canada. So I got the plants from a person, and when I got them, they were already contaminated because he had created his own system, and all the brassicas just happened to get this disease. It's a really big problem in Canada. And so when I got them, they had them, and there isn't a lot of research done on how to get rid of the disease. And so David and we started to experiment with how to get rid of them. So I used a fungicide in the media, and then usually with this fungus comes a bacteria. And so we did an antibiotic on it as well, and then it resulted in no chloroplast. So I don't know.
So that's why it's completely white.
Yeah. So
they look kind of cool though.
They do. They look awesome.
But they probably couldn't survive outside of tissue culture.
But I'm gonna transfer them over and just keep them alive to see how this turns out. I'm getting new plants in September. I'm getting a bunch of plants from X-Plant Labs. So I know what to do now that I did wrong the last time.
Awesome.
Yeah.
The principle of cryopreservation is preserving material under ultra low temperatures. So think about, let's say you get a batch of seeds, right? Usually most people work with seeds, they will put them in an envelope and store them in the refrigerator, which is fine. And that prolongs the lifetime of the seeds. So if you want to germinate a seed months later, it will be still viable, but not always. A lot of those seeds, they lose viability over time. Cryopreservation allows you to store them under these ultra low temperatures and basically stop all the metabolic activity in the seeds. By stopping that, you're halting their deterioration or viability loss. Mm-hmm. So you can maintain a high viability. How do we do that? We prepare certain solutions. There's a process called vitrification, which is, in very simple terms, the cells in the seeds, they have water inside. You don't want that water to freeze, because the water freezes, what happens? It forms ice crystals. Those ice crystals will cause damage to the cell walls. So in order to prevent the ice crystals from forming, we use solutions that basically enter the cell, and they are highly concentrated, and they create almost like a super cool gel. It never freezes. So those are called, the system is called vitrification. It has this kind of vitreous, kind of glass looking, but it never forms ice crystals. So that preserves the integrity of the cell. So by using the solution, and then you put them under liquid nitrogen, which is minus 196 degrees Celsius, you can preserve the seeds indefinitely, forever. When you're ready to use them, you take them out, you thaw them in warm water very quickly for a minute. You remove that solution through a filter. You wash those seeds, and then you put them in a Petri dish with a culture medium, and they would germinate. The process cryopreservation can be a little harmful because of some of the chemicals used in the solution. When you're working with seeds, you check the viability of the seeds using some tests, like the tetrazolium test is one. So let's say you determine that your batch of seeds has a viability of 96%. When you do cryo, if you can get at least half of that to recover, let's say 48% in this case, that's considered successful. With the vitrification solution, it's called pre-vitrification solution, and there are several. There's 1, 2, 3. We usually use the pre-vitrification solution 2, PVS2. Because of the some of those components are harmful, when we take them and thaw them, we need to thaw very quickly and wash them with a culture medium, for example, MS, liquid medium. We just wash to remove the excessive chemicals of the PVS2 solution, and then we put them for germination. And then we do an evaluation of the germination. So let's say if we get a germination of 50% and the initial was 96%, that's considered success. Mm-hmm. If it's lower than 40%, it's still good, but it's not successful. But by tweaking the chemicals, we found out, for example, by using fluoroglucinol, which is another chemical, we can increase that recovery. So instead of getting 48%, we can get close to 92%.
Wow.
So very close to the initial viability. So we work different protocols that we can get really successful results. And why is that important? Because orchids are considered endangered. It's important to preserve that seed material for the future, for if you want to germinate them in the future, or if you want to send them to a botanical garden elsewhere in the world, mm-hmm. or for conservation purpose. Now we can cryopreserve not only seeds, but pollen, also tissues in vitro, like somatic embryos or in vitro cultures.
Do the somatic embryos, you said the seeds last indefinitely, will the embryos also last indefinitely?
Yes, in theory, the process cryopreservation preserves any material indefinitely. That's the premise of cryopreservation. You can keep it there forever until you need it.
We had worked in the same lab when we were in graduate school at different
really?
times.
Wow, that's crazy.
And I worked with some plants that he had put into cryogenic storage, which would have been probably at least 15 years before or so. And we took them out, and some of them were not terribly viable, but the ones that had been handled carefully and that we worked with, they all came out, and we were producing plants from them within a month or so.
Commercial labs, usually they will have a big or 1 or 2 tanks of liquid nitrogen attached to a big storage tank. What you saw there is a small tank, but they will have huge ones with a line that feeds automatically the liquid nitrogen, and it has an alarm that tells you when the levels are getting low, so you can always keep replenishing it.
We had been keeping it here because we're bringing them in and out of storage, and so we can wheel it over. So you do not want to touch the metal when it's in here. Obviously it's going to be somewhat cold. So you can see we put the samples in a box in here. The boxes have to be ventilated because or else they would fill with the liquid nitrogen, and then when we took them out they would blow up. I don't have one sitting right here, but you can see it's obviously fairly cold.
Those sci-fi movies, when people get cryogenically stored, and then they come back to life, we do that with the plants.
That's awesome.
We can do that with seeds, with pollen, with embryos, somatic embryos, with anything basically.
This is the cryo storage tubes. You have to use a very small sample regardless of what you're using, because the larger the sample, the harder it is to cool at the right speed. And if it cools at the wrong speed, ice will form, and when the ice forms, it will destroy the cells.
That tube has a volume of 2 milliliters, but you can probably have there easily 2,000 seeds in that tube
Wow.
because they're very tiny. Yeah, it all ties up. You know, when you think about micropropagation, it's not only about multiplying plants, but there is a purpose for that. Why do you do tissue culture? Yeah, commercially, you want to do mass clonal propagation of a plant that is unique, that you want to sell maybe, or you have a rare plant, endangered, that there are only a few species left. You can use tissue culture to multiply them and maybe reintroduce into native habitats. You can use that system for producing pharmaceuticals, secondary metabolites. So there are all sort of applications. So it's a nice field. I would say you're never bored when you work with micropropagation.
I hope you guys enjoyed the tour. Thank you so much to Dr. Vendrame and David for lending me your time. Pretty much everything that I know about tissue culture, I learned either from reading books or from watching videos about it on the internet. So it is really special when I get to talk to people who work and research in the industry and ask them all the questions that I have. Bye!